Hi, my name is Sam Naiman, and I'm here to talk to you today about a paper I recently published in PNAS on genomic rearrangements and local adaptation. And really what I'm interested in with this paper is trying to explain the occurrence of genomic islands, which are clusters of locally adapted loci that occur in the genome. And I'm trying to answer two questions here. Is divergence hitchhiking important in generating these islands of divergence? And if not, can these islands evolve through the spread of chromosomal rearrangements? Now, the general reason for looking at this is that there's an effective recombination on local adaptation. And if we kind of conceptualize local adaptation as occurring with chromosomes and locally adapted alleles, so either two red or two blue alleles, if there's a crossover between these alleles on the same chromosome, we get a mixed up gamete that will have a red and a blue allele. This will break up this locally adapted complex, this combination of two good alleles. Whereas if we have loci that are tightly linked on the chromosome, recombination won't tend to break them up as easily, and we'll maintain this combination. This general disadvantage of recombination has been studied for a very long time in the context of local adaptation and other similar processes as one force that may be kind of shaping the genome or shaping the way that things adapt locally. One of the main results that I present in this paper is about the relative advantage of being tightly linked to an existing locus that's locally adapted compared to a new mutation occurring anywhere else in the genome. And here we're looking at the establishment probability of these new mutations. And so the divergence hitchhiking theory suggests that because you're tightly linked, you should have a higher establishment probability. And this graph just quantifies how much greater that advantage should be than being unlinked. And what it turns out is that over most regions of parameter space, over wide ranges of migration, this is a relatively small advantage. There's not much advantage to being tightly linked compared to occurring anywhere else in the genome, except for in these very restricted regions of parameter space. So the take-home message from this part of the paper is that this relatively small advantage of divergence hitchhiking won't result in a very strong signature of local adaptation or of genomic islands because the genome is a very large place. For every mutation that does occur in a cluster, there will be lots of mutations that establish throughout the rest of the genome. And so we won't expect to see a very strong statistical signature of genomic islands due to this divergence hitchhiking mechanism. I then turn around and look at instead what happens if we have rearrangements bringing loci from different parts of the genome into tight linkage. This graph here shows the results of individual-based simulations that have rearrangements. So what we have is we have time on the x-axis from 0 to 500,000 generations, so deep time. And on the y-axis, we're looking at along a chromosome from 0 to 50 centimorgans. And we have loci in blue here that are contributing to this locally adaptive phenotype. On the left, we have a homogeneous environment where both patches in this two-patch model are in the same type of environment. And on the right, we have a heterogeneous environment. What you can see is there's very little change in the arrangement of these loci on the chromosome in the homogeneous environment. But in the heterogeneous environment, we get very, very rapid and clustered, highly clustered architectures evolving over this deep time. And you can see here all of the loci involved in local adaptation have rearranged to be in this tightly linked cluster. I spend a lot of the rest of the paper looking at the different parameters and the effect of different parameters such as migration rate, population size, and rate of rearrangement on on how fast you expect to get these clusters of locally adaptive mutations or locally adaptive loci evolving over time. Basically, the take-home messages of this paper are quite simple. I don't really expect divergence hitchhiking to be generally very important, and it does seem that genomic islands can evolve through these rearrangements, given that the population size is large enough and the rate of rearrangement is high enough, and we do have these kind of heterogeneous environments. Of course, these are all empirical questions, and it'll be really interesting to see whether we find these patterns in nature. Also, really important will be for people to start studying and trying to tease apart whether the loci that are actually involved in these genomic islands are in fact contributing to local adaptation or just neutral loci along for the ride. So these are really cool empirical questions, and I'm really excited to see what people find. Please get a hold of me with your data. I'd love to see it. Thanks for watching.